Hello, everyone. In this module 5, I will introduce the clustering and talk about hierarchical clusterings, which is a basic but fundamental concept for the unsupervised machine learning algorithm. And the next module, we will cover other clustering methods such as k-mean clustering, mixture models, and model-based clustering. Here we go. Previously, we learned a few supervised learning methods such as regression and random forest. In the supervised learning setting, we typically have access to a set of features, x1, x2, and xp, measured on n observations, and a response y, the response variables, also measured on those same n observations. The goal of the previous modules was to predict y using x1, x2, and xp. But for this model, we will focus on unsupervised learning. Unsupervised learning is a set of statistical tools intended for the setting in which we have only a set of features, the predictors x1, x2, and xp, measured on n observations. We are not actually interested in prediction because we do not have an associated response variable y. And the goal is to discover interesting things about the measurements on x1, x2, and xp. So the main question is that, can we discover subgroups among the variables or among the observations? Through this module, we will focus on clustering, a broad class of methods for discovering unknown subgroups in data. So clustering. So cluster analysis divides data into groups or clusters that are meaningful, useful, or both. If meaningful, then clusters should capture the natural structures of the underlying data generation process. Alternatively, clustering can be useful for summarizing or reducing the size or complexity of the data, similar to PCA. And please remember that clustering involves automatically discovering natural grouping in data. Unlike supervised learning, clustering algorithms only interpret the input data and find natural groups or clusters in feature space. There are two important views of clustering. First, find homogeneous subgroups. Partition data into groups such that objects in the same groups are similar to each other, while objects in different groups are dissimilar. Second, statistical clustering. Now, partition data into groups such that objects in the same groups are from the same distribution. Here are 4 fields of clusterings. Please stop and read them through. And please remember that there are probably more approaches and algorithms for clustering than any other areas of data mining. In my opinion, there are 3 most popular methods in which are the building blocks of the more complex and targeted method. The 3 most popular methods, in my opinion, are hierarchical clustering, k-mean clustering, and model-based clustering. Today, we will cover hierarchical clusterings first and learn other methods in the next module. First off, we will learn in the next module that one potential disadvantage of k-mean clustering is that it requires us to pre-specify the number of clusters. So hierarchical clustering is an alternative approach that does not require that we commit to a particular choice of k. In addition, hierarchical clustering has an added advantage over k-mean clustering, in that it results in an attractive tree-based representation of the observation called the dendrogram. So hierarchical clustering creates a set of hierarchical clusters based on a dissimilarity distance metric. The resulting structure is represented by a dendrogram tree drawing, which resemble a tree. Let's start with the dendrogram. Here, I generated 45 random points, and it can be seen from the naked eye that the data points from 3 clusters. Let's check the denote script. Suppose we have collection of data points represented by numpy array x here, which are generated from 3 different normal distribution. And the script here draws the data points in the x and labels the data points from 1 to 45. In the image below here, we can see the points that is generated from this code block. Let's go back to our slide. So it can be seen from the naked eye that the data points from 3 clusters, the first at the bottom left consisting of points 1 to 15, second at the top right consists of points 16 to 30, and the third at the bottom right considering points 31 to 45. However, please remember that in the real world, we may have thousands of data points in many more than 2 dimensions. In that case, it would not be possible to spot clusters with the naked eye. This is why clustering algorithms have been developed. In this module, I will describe bottom-up or agglomerative clustering. This is the most common type of hierarchical clustering and refer to the fact that a dendrogram is built starting from the leaves and combining clusters up to the trunk. From the denote here, use this code block and you can draw the dendrogram for our data points. Here I use the SciPy library for the purpose and execute this script, and you can draw the dendrogram for our data point here. Let's go back to our slide. We will begin with a discussion of how to interpret a dendrogram and we will begin with a discussion of how to interpret a dendrogram and then discuss how hierarchical clustering is performed. That is how the dendrogram is built. Basically, the number of clusters highly depends on where you cut the trees. These example figures show the variance of the cluster results. So different color here indicate different clusters. There are many methods for grouping the data. Thus, we will learn 5 common linkage methods in this module, and we will also learn how we can choose the right number of clusters. So we have 2 clusters here, and we have 3 clusters here, and we have 6 clusters here. So the hierarchical clustering is based on the distance between each point. For example, point 1 and 2 are very close compared to point 3 and 9. Thus, point 1 and 2 are very likely to go into the same cluster, while 3 and 9 are likely to be separated. There are 5 linkage methods to calculate the distance between groups. So the basic hierarchical clustering algorithm takes a greedy sequential approach to forming the nested groups. Actually, the hierarchical clustering dendrogram is obtained via a extremely simple algorithm. This is how the agglomerative hierarchical clustering algorithm works. Please pause the video and try to understand these 5 steps. And the algorithm proceeds iteratively, starting out at the bottom of the dendrogram. Each of the n observation is treated as its own cluster. And the 2 clusters that are most similar to each other are then fused so that there are now not n-1 clusters. Next, the 2 clusters that are most similar to each other are fused again so that there now are n-2 clusters. The algorithm proceeds in this fashion until all of the observations belong to one single cluster and the dendrogram is complete. So the start from n cluster, for example, here we start from 9 clusters, and we can begin by defining some sort of dissimilarity measure between each pair of observation. So most often, Euclidean distance is used. So we will discuss the choice of this similarity measure soon. as well. And after one iteration, we have 6 clusters, as shown here. After the third iteration, we have 3 clusters, and after fifth iteration, we have 2 clusters. This is basically how the agglomerative hierarchical clustering works. And this algorithm seems simple enough, but one issue has not been addressed. Consider these 3 clusters here. So how did we determine that the cluster 1, 2, 3 should be fused with the cluster 4, 4, 5, 6? So we have a concept of the dissimilarity between pairs of observations, but how do we define the dissimilarity between 2 clusters if one or both of the cluster contains multiple observations? So the concept of dissimilarity between a pair of observation needs to be extended to a pair of groups of observation. This extension is achieved by developing the notion of linkage, which defines the dissimilarity between 2 groups of observations, not the point. So the 5 most common types of linkage are single, average, complete, centroid, and wide linkage. Now let's see common methods to calculate the dissimilarity between clusters. So the first 3 are based on the pairwise dissimilarity or similarity between observations. Let A and B be 2 clusters, the collection of points, and Dij is the dissimilarity distance between observation I and J. I will introduce basic but commonly used linkage method. The first one is single linkage or nearest neighbor. The second one is complete linkage or farthest neighbor, and the third one is average linkage. So the single linkage are most popular among statisticians. The single linkage computes all pairwise dissimilarity between observations in cluster A and the observation in cluster B and record the smallest of these dissimilarities. The single linkage can result in extended trailing clusters in which single observations are fused one at a time. This is the minimal intercluster dissimilarity, we call that. However, average and complete linkage are generally preferred over single linkage as they tend to yield more balanced dendrogram. And the complete linkage computes all pairwise dissimilarity between the observations in cluster A and the observation in cluster B and record the largest of these dissimilarity. This is the maximal intercluster dissimilarity. And third one, the average linkage computes all pairwise dissimilarity between the observation in cluster A and the observation in cluster B and record the average of these dissimilarities. So average linkage is the mean intercluster dissimilarity. And another common approach is to base the cluster dissimilarity score on the centroid and compactness of the observation in the cluster. The first one is centroid linkage and second one is wide linkage. The centroid linkage first, it calculates the dissimilarity between the centroid for cluster A and the centroid for cluster B. However, centroid linkage is often used in genomics but suffer from a major drawbacks in that an inversion can occur, whereby 2 clusters are fused at a height below either of the individual clusters in dendrogram. This can lead to difficulties in visualization as well as in interpretation of the dendrogram. The last one is wide linkage. Personally, I prefer wide linkage over other methods. So the idea has much in common with knowledge of variance, the ANOVA, and the linkage function specifying the distance between 2 clusters is computed as the increase in the error sum of square after fusing 2 clusters into a single cluster. Wide linkage uses the dissimilarity score as shown here. Thus far, the example in this module have used the Euclidean distance as the dissimilarity measure, but sometimes other dissimilarity measures might be preferred. For example, correlation-based distance considers two observations to be similar if their features are highly correlated, even though the observed value may be far apart in terms of Euclidean distance. So the choice of dissimilarity measure is very important as it has strong effect on the resulting dendrogram. In general, a careful attention should be paid to the type of data being clustered and the scientific question at hand. So this consideration should determine what type of dissimilarity measure is used for hierarchical clustering. For our key example, I will use the purple rock crab data. This data includes 5 morphological measurement on 200 crabs from the species purple rock crab. The purple rock crabs can take on a blue or orange color in some parts of Australia. In the dim note script, you can find these code blocks. If you change the color here, the color red value, you can check how the number of clusters, the K value changes with respect to the different height values. So one very important question here is, what is the right number of clusters, the K? There are many approaches for selecting the number of clusters. One approach is to look for regions in the dendrogram where gaps or change appears in the height of merges. The height corresponds to the dissimilarity between the merged clusters, so a large jump in height corresponds to the high dissimilarity, right? So between the solution for K and K plus 1 clusters. And for Ward's method, this corresponds to the change in the sum of square error, the SSA for merge. For complete linkage, the height corresponds to the dissimilarity between the most dissimilar points in the two clusters. So as you can see from this figure, you may choose the region here, which shows a very large gap in the height of merges compared to other regions. In addition, you can actually check how the number of clusters changes with respect to the height. So here are a few more essential aspects that you may want to study more in the future. First, please remember that a choice of dissimilarity distance can be crucial for the clustering result. Second, the predictors might need to be standardized. And third, some features might be more important than other features. Thus, we may need to give some weight to some features that you think more important when we calculate distance metrics. And fourth, you may also want to consider TCA. And fifth, please remember that there are many linkage methods. So you need to decide which linkage method you should use and why. And sixth, as we learned, we need to decide what value of K to use. And finally, please remember that clustering methods are not perfect and do not usually lead to repeatable patterns. This is the end of the slide for this module. Thank you.